membrane transport. Let's call it that. Membrane transport. So we're talking about the fact that obviously things have to move in and out of cells, and how does this happen? And it happens in many different ways. So we'll just quickly talk about diffusion. Diffusion does not necessarily require a membrane. Diffusion is just the movement of anything from a high concentration to a low concentration, right? High to low. So with concentrations, it's important to think about them as gradients or slopey lines. So let me talk a second about gradients. Yeah, you guys kill me. All I wrote was that. There you go. So it's important to think of gradients. And then the best way to think of gradients is to imagine the most common gradient that we're familiar with. And that, of course, is what we call a hill, right? So when we're talking about a gradient, maybe this is sort of like an aside here for concentration. When we're talking about a concentration gradient, well, we could talk about a gradient of dirt. So here's a gradient of dirt. If you take a flat space and you start to shovel, dig a hole, and you pile the dirt up over here on the other side, and you keep digging, right? And you keep digging, so I'll use my eraser here and I'll dig a hole over on this side. Down we go. And then I'm piling my dirt up over here to make a pile. Well, eventually, what we end up building is a difference in height. That's what a gradient is. It's a difference. And if I did a really good job and smoothed it all out really nice, I could have a nice gradient. Here's sort of like where the original ground was. But now I've established a definite low place and a definite high place of dirt. And this is called a hill. This is the hill of dirt. But it's possible to have a hill of concentration. You can have a hill of charge. In physics, we talk about voltage gradients. We're going to talk about that also in biology with nerve cells and how they can create a hill of electric charge, a place that's higher and a place that's lower. And of course, with regular hills of dirt, if you put a ball on a hill of dirt, it rolls down sort of all by itself. It doesn't require any energy. No one has to push it. No one has to do anything to it. It just automatically goes. It's always trying to go from high to low, right? Well, the same thing is true for things that are on a concentration gradient. They want to go from places where it's high, and they want to sort of roll down the concentration hill. And that's what makes them go. If you want something to go up the hill, you got to push it. It won't go by itself. If you want to go against the gradient from low to high, you're going to have to push somehow. And so when we're talking about concentrations, if we want to move something from a place where it's low concentration and force it to where it's high, we're going to have to push it. It won't go on its own. That's the idea. So a concentration gradient works very much like a hill. What's interesting about the word gradient is that it's also called a slope, right? And gradients can be shown on graphs as slopes. And so all these words, math words and science words, they all sort of mean the same thing. Okay, so that's what diffusion is. And diffusion happens anywhere you have concentration gradients. Mother Nature is always trying to work towards equilibrium, where everything is balanced. And the reason for that is because the random bustle and jostle of molecules is pushing on each other. And if you have a crowd, then they push and they tend to disperse. Just like people. If you put a bunch of people in the outside parking lot and you crammed them all together and just waited, well, eventually, as they're talking and moving and doing their thing, they would slowly begin to spread out, right? Because nobody wants to be packed in that tightly. And so it's the same idea. The people would diffuse through the parking lot and spread out and take more space. Now, when you put a membrane in the middle and separate two spaces, that's when everything gets a little bit complicated. So if we take a membrane and we separate two areas where there's a high concentration on one side and a low concentration on the other, this is a hill. This is a gradient of high to low, which means if the membrane wasn't there, the particles on the left would try to migrate towards the right. They would want to roll down the concentration gradient hill. Problem is is that they can't if the membrane in there prevents them. This is what we would call a selectively permeable membrane. So only certain things can go through it. And so if these black little dots cannot go through the membrane prevents the equilibrium from occurring. It prevents this balancing out. But that's where something cool can happen. Water, which is the solvent all around here, is in a very large amount on this side. The proportion of water to solute is quite high. Whereas over here, because of all the black solute, there's actually less water, right? The proportion is much greater. So the water itself can seep through the membrane and go from the low concentrated part to the high. Now, it's still a concentration gradient if you think of it in terms of reverse. Normally, we think of water as the solvent, and the black dots are the solute, right? But they're just molecules. So on the right side, there's way more water than on the left. You could argue that there's a concentration gradient of water. That's the opposite of the concentration gradient of the black dots. This is high concentration of water. Now, that doesn't really make sense because the water isn't in a solvent. It is the solvent. So it's not a very accurate way to describe what's actually going on, to say there's a high concentration of water. That doesn't really make sense. But in a way it does, right? There's more water available on that side. There's more water in proportion. And so the water will move across the membrane in the process of osmosis. So osmosis can only happen if you have some kind of membrane that is preventing the movement of solute in one direction, from high to low, and then what happens is the water, if possible, will seep through the membrane the opposite way and move from what we would call the low to high concentration of solute. That's osmosis. Okay? Now, in terms of cells, there are lots of little molecules that can cross a membrane by simple diffusion. They can just work their way through. And so by diffusion, we often see very small molecules, things like oxygen, carbon dioxide, water. These things can typically slide right through a membrane without too much trouble because they're very small. And the membrane molecules are very large and bulky. So it would be kind of like, imagine a jar full of large marbles. Well, if you put tiny bits of sand in there, the sand can easily seep between the marbles, can't it? So that's kind of the idea here. So basically, small molecules can diffuse right through a membrane without too much trouble. The other thing is, of course, you can also get, if you have non-polar molecules, because the membrane is made of fats, right? The middle of the membrane is the non-polar tails of those lipid molecules. Non-polar molecules are more capable of sliding right through a membrane than polar ones. Even if they get a little bit big, they can still slip through. We'll see that later. But typically, most membranes are very selective in what they let through. So larger molecules, biological molecules like lipids and carbohydrates and the things that we are talking about are generally too big to slip through a membrane on their own. And so what happens, of course, is we have to transport them across the membrane. And the way the membrane does that, is by utilizing the protein gates. Okay, the protein gates. So I'll draw a little protein here, globular, goofy protein on the other side of my membrane. There, something like that. And so what can happen is this protein molecule, its shape can be altered to create a channel that can open. And so if we alter the shape of this protein such that a tunnel forms in the middle, then solute that was normally over here and wanted to go, now it can flow through the channel. And this is called facilitated diffusion. Oops, I spelled that wrong. I keep spelling that word wrong. just a C in there. Facilitated diffusion. In other words, the cell is being helped, or the solute is being helped. It's still diffusion. It's still going from a low concentration to a high concentration. A high concentration to a low concentration, right? It's still following the gradient. It's doing it automatically. It's rolling. Essentially, what we're doing is we're taking this permeable membrane, we're poking a hole in it, and allowing the solute through. Now, how do you get the protein to do that? Well, there's a number of different ways. One of the ways is if the concentration on this side builds up too high, then the interaction of this high concentration can affect the shape of the protein and cause it to open the channel. There are other ways to open these channels, including voltage differences, and also what we call ligands, where other molecules will bond to the protein and change its shape. And we're going to get into that in more detail later on. But facilitated diffusion is still regular, passive, rolling down the hill, but it's happening through the protein pores or gates that have opened up. So far, so good. Now, another thing that cells must do is they must also sometimes push things up a concentration hill. This molecule might be something that is detrimental to the cell, or that it needs to move, or that it needs for life. It might be glucose in the blood that it wants to grab because it needs it. So we want to get this to move this way. But of course, that's going low to high. So you can't do that just by opening a hole. It won't work. If you just open a hole, it'll actually go this way and go the wrong way, right? So how do we get this to happen? And this is what we call active transport. Active transport is basically going to cost us something. The passive transport, because it's rolling downhill, happens. All we got to do is open a hole and it'll just go. But active transport means we have to push it against the concentration gradient. So we have to push it up the concentration hill. And that requires energy. And the typical form of that energy is the molecule ATP, adenosine triphosphate. And what adenosine triphosphate does is it uses the energy in its bonds. It's basically a molecule of adenosine and 3 phosphate groups. It's a nucleotide, and it has 3 phosphate groups attached to it. And sometimes it can have 2. When it has 2, it's called ADP. And sometimes it can only have 1, and it's called AMP, adenosine monophosphate, adenosine diphosphate, adenosine triphosphate. All we need to remember right now is the more phosphates it has attached, the more energy it contains. And by dropping a phosphate off, it can transfer chemical energy to something else. So what typically happens, and that's called phosphorylation, that process where you add phosphates to things. So if we go back to our membrane, and we have this protein molecule in here, like this, one of the things we can do is force it to open a channel. But the way we do it is slightly different. In this case, an ATP molecule has to be floating by on one side. And what it does is it hands off a phosphate. It attaches one of those phosphates to the protein and then becomes ADP. And by doing that, it attaches this phosphate, which then, of course, changes the shape of the protein molecule, and then that causes the channel to open. All right? Now, it's not just as simple as a channel. What actually happens is it's more like a grabbing of the molecule. So let's go, this is our high concentration area, and this is our low concentration area. It's more like the changing of the molecule allows one of these things to be grabbed and then sort of squeezed or pushed through the protein molecule, right? Opening a channel isn't enough because it won't flow. It won't go on its own. You have to kind of grab it and almost like squeezing a tube of toothpaste, you know? You start at one end and you have to squeeze it through to get it over here. And it's all about the shape of that protein molecule and the phosphorylation, the adding of the phosphate. Anyway, because the ATP represents energy, this means that we are using energy to make this happen. That's why it's called active transport. It costs us. And that energy, where do we get the ATPs? Well, the process of glycolysis and respiration is the process that makes ATPs out of glucose and oxygen, right? So our body's making that ATP just for the purpose of doing this. So now it can use the ATP to pull things in and out of cells in active transport. And that's sort of the idea of, or the difference between those three different kinds of transport. There are many different examples of active transport, and rather than go through them all now, we're going to go through them as they become important. One of the most common is called the sodium-potassium pump. So what I want you to do on the weekend is I want you to look up the sodium-potassium pump and just kind of get an idea as to how it works. And just read through that. So when you come back on Monday, I'm going to ask you questions about the sodium-potassium pump. Maybe it'll be a quiz. It'll be like, ooh, I'll have to mark it. And you'll be scared. That's the only way I can force you to actually do it. Because you're not motivated enough to learn it because it's interesting and because it's important in your biological future. But you are motivated if I say, maybe I'll give you a little quiz on the sodium-potassium pump. And you'll be like, oh my God, I better read it. So there'll be a very short quiz on the sodium-potassium pump on Monday. So you have to read about it. You have to basically learn as much as you can about it so you can be ready for whatever quiz the professor decides to hammer you with. Right? This is year one university we're preparing for. So that's how they do it. They don't say, learn these 5 things. They say, study this, and I'm going to test you on it. And you're like, well, how much do I study? And they're like, shut up, study. Right? So learn it as much as you think is reasonable, and we'll see how well you do. It could be out of 1 mark. It could be 1 mark, and each point is worth 0.07. It doesn't matter what it's out of. It's all in the value. So, and again, professors don't tell you that either. Professors just say there's a quiz or there's a test. Then that's all they tell you. So study what you can, learn what you can. Obviously, this is not going to be a killer thing where I'm going to go nuts and hammer you. I'm just doing this to get you to learn this. So it's probably going to be a relatively basic little quiz. All right, good enough. We'll stop there.